Biofilm formation in water piping system occurs when microorganisms such as bacteria, fungi and algae attach to the inner surfaces of pipes and form complex communities. These microorganisms produce a sticky substance called extracellular polymerase substances EPS, which help them adhere to surfaces and protect themselves from environmental stress, including disinfectants. In water system, biofilm formation is influenced by factors like water quality, temperature, nutrients, and flow rate. Over time, these biofilm grow and mature, creating a habitat for various microorganisms, including harmful ones like Legionella, Pneumophila, and Pseudomonas aeruginosa. This biofilm can cause problems like reduced water flow, pipe corrosion, contamination, poison health risks, and operational challenges.
Biofilm are interactive communities of microorganisms that stick to surfaces and are encased in an extracellular polymeric substance EPS matrix that they have a self-produced water piping network and other natural and artificial habitats are replete with this microbial population. Biofilm formation is a major concern in the context of water distribution networks because of the possible threats it poses to infrastructure integrity and water quality. Microorganisms like bacteria, fungi, algae, and protozoa adhere to the inside surfaces of water pipes and start to multiply, creating thick layers that eventually form biofilms. The creation of persistent microbial reservoirs that are challenging to remove. Pipe corrosion and drinking water contamination are just a few of the major problems that this biofilm can cause. There are many different types of microorganisms that can produce biofilms in water piping system, but bacteria are the most common. Some of these species are dangerous and can lead to major health problems, such as Legionella, Pneumophila, Escherichia coli, and Pseudomonas aeruginosa. An example of an opportunistic pathogen that is frequently found in biofilms inside water system is P. aeruginosa, whereas L. P. Mopila is responsible for the severe form of pneumonia known as Legionnaires' disease. A variety of fungi, protozoa, Mycobacterium avium, and Staphylococcus aureus are among the other bacteria that can form biofilms, adding to the complexity and diversity of microbial communities in these settings.
Okay, the next one is biochemical pathway of biofilm formation. The biofilm formation is a sophisticated process that involves a series of biochemical pathway and regulatory mechanisms. This pathway enable microorganisms to adhere to surfaces, communicate with one another, and develop complex community structures. This is a detailed exploration of the key biochemical pathway and mechanism involved in biofilm formation. In this slide shows you two different metabolism in biochemical pathway of biofilm formation. And the first one is glycolysis and energy metabolisms, and the second one is amino acid metabolisms. The glycolysis is the primary pathway for energy production in many bacteria, providing ATP and metabolic intermediates essential for growth and biofilm formation. In this metabolism have two different key enzymes, which is phosphofructokinase and glyceraldehyde 3-phosphate dehydrogenase. The first one is regulates the commitment step of glycolysis and is crucial for energy production. And the second one is to convert glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, generating NADH, which is vital for cellular respiration. Lastly, about the biofilm implication in this metabolism, to enhance glycolytic activity, support the rapid growth of planktonic cells, facilitating initial attachment to surfaces. Once attached, cells can switch to different metabolic states that favor biofilm development. And the second one is amino acid metabolisms. Amino acids serve not only as building blocks for protein but also as critical metabolites that influence biofilm formation. The key pathway in this metabolism is amino acid catabolisms. This pathway that degrade amino acid produce various metabolites that can enter central metabolic pathway. And the second one is shikimate pathway. The pathway is responsible for the synthesis of aromatic amino acid, and it's linked to biofilm formation certain bacteria like Pseudomonas aeruginosa. And the last but not least, biofilm implication in these metabolisms. The availability of specific amino acid can enhance biofilm formation. For example, species Maltophilia show a preference for amino acid over glucose, indicating that amino acid metabolism may be more favorable for biofilm development under certain conditions.
Besides that, extracellular polymeric substances, EPS. EPS are crucial components of biofilms, providing structural integrity and protection to microbial communities. The composition of EPS are: EPS primarily consists of polysaccharides, proteins, lipid, and extracellular DNA. The exact composition can vary significantly between species and environmental conditions. Biofilm implication for EPS is EPS not only protect bacteria from environmental stress, but also facilitate intercellular communication and nutrient trapping. The production of EPS is often regulated by metabolic state and environmental signal. Next, quorum sensing. Quorum sensing is a cell-to-cell communication mechanism that enables bacteria to sense their population density through signaling molecules known as autoinducer. The key components for quorum sensing are first, autoinducers. These signaling molecules accumulate in the environment as bacteria population grows. When a threshold concentration is reached, they trigger changes in gene expression. Second, regulatory genes. Genes involved in biofilm formation are often regulated by QS system. For example, last L and last R in Pseudomonas aeruginosa. Biofilm implication for quorum sensing is QS regulated the expression of gene responsible for EPS production, virulence factors, and other processes critical for biofilm maturation. The disruption of QS can lead to reduced biofilm formation and increased susceptibility to antimicrobial agents. Last but not least, metabolic switching. During biofilm development, bacteria can switch from a planktonic lifestyle to a sessile lifestyle characterized by altered metabolism. There's two key aspects of metabolic switching, which is first, metabolic reprogramming. As bacteria transition to biofilms, they often downregulate pathways associated with rapid growth and upregulate those associated with survival and persistence. Second, central metabolism adjustment. The shift may involve increased reliance on fermentation or alternative carbon sources when oxygen levels are low or when nutrients are limited. Biofilm implication for metabolic switching This metabolic flexibility allows bacteria to adapt to changing environmental conditions within the biofilm matrix, enhancing their survival under adverse circumstances. So, the biochemical pathway involved in biofilm formations are intricate and interdependent. Understanding this pathway provides insight into microbiome behavior in natural environments and clinical settings. Targeting specific metabolic processes or signaling mechanism could offer novel strategies for disrupting biofilms associated with persistent infection or industrial fouling. Continued research into this pathway will deepen our understanding on microbial ecology and inform the development of effective antimicrobial therapies. There's a diagram of biochemical pathway of biofilm formation in water pipeline system.
Today, I would like to present about introduction of case study. Biofilms. Biofilms are group of microorganisms like bacteria, fungi, and algae that form on surfaces submerged in water, such as pipes and filtration systems. These biofilms are embedded in a self-produced substance that protects them and helps them stick to surfaces. In water pipes, biofilms can lower water quality by introducing harmful pathogens, causing corrosion and affecting taste and odor. They also promote blockages and increase maintenance costs. Biofilms form when microorganisms adhere to the surfaces of pipes and grow, creating a protective layer that harbors dangerous pathogens like Legionella and E. coli. While water treatment plants provide clean water, the quality can decline during distribution due to physical, chemical, and microbial stresses. Preventing biofilm formation through disinfectants, proper pipe materials, and maintenance is crucial, especially for vulnerable groups like infants, the elderly, and those with weakened immune systems. The study emphasizes the need for strategies to monitor and control biofilm formation to ensure safe drinking water.
Case study overview biofilm formation in water distribution system. The paper provides an overview of biofilm formation investigation and control, focusing on drinking water distribution system. It updates novel method for bacterial biofilm analysis and summaries recent discoveries and chlorine resenses impact on biofilm formation. The study also examines the material in drinking water network and their related to biofilm development. Biofilm can be found in nature in living organisms in industry environment in the water distribution system. In the food industry, the presence of biofilm layer on product surface and equipment can be significant hygiene problem. Biofilm can be also be formed by pathogenic microbe, which can cause not only industry hygiene problem but also serious epidemiological problem. Biofilm in drinking water distribution system cause different problem in water quality, discoloration, change in the odor and taste and safety. There are many definitions of biofilm in the literature, but they all essential stage a very similar terminology, according to which a biofilm is a biological active matrix attached to the surface in cell in their extracellular product. EPS, extracellular polymeric substances, microorganisms not only adhere but also multiply, and the organic polymer they produce play a role in the formation of biofilm. EPS formation depends on the composition of the matrix from forming microbe and their physiological stage, as well as the available nutrient. Metabolism change with the biofilm, with anaerobic microorganism close to the surface of aerobic microorganism. In the aqueous burst, with the matrix, there are capillaries and channel touch which the cell absorb nutrient.
Biofilm formation in general has been frequently reported in the literature, and the stage and process of biofilm formation in DWDSS occur in a similar way. Microorganisms prefer to attach to a solid surface because it provides greater protection and a better supply of nutrients than the plant technique stated. Biofilms are more likely to form in flowing, nutrient-poor systems, example gratia, a drinking water distribution system, because the starving cells can obtain a continuous supply of nutrients by adhering to the surface. Fleming and Cavaders reported that over 95% of the biomass in DWDSS exists in the form of biofilm, with only 5% detectable in planktonic form. During biofilm formation, proteins, carbohydrates, and other organic or inorganic molecules rich in nutrients adhere to the surface of equipment and piping system, and by diffusion or turbulence flow a conditioning film is formed, which changes the electronic charge and hydrophobicity of the surface in a way that is beneficial to the biofilm. If a flowing aqueous phase is present in the system, the nutrient supply of the conditioning layer is much better than of the aqueous phase. Once the conditioning layer is formed, cells can adhere, but this depends largely on the nutrient compositions and other environmental parameters. In some microbes, different proteins promote adhesion. For example, according to some studies, albumin promotes the adhesion of Listeria monocytogenes, but inhibitory effects can also occur. For example, Hack and Cavaders also reported the inhibitory effect of casein on Listeria monocytogenes. The formation of the conditioning layer is also strongly influenced by the material and roughness of the surface. It also provides a protective function against adverse physical and chemical, environmental effects, dehydration, thermal effects, mechanical cleaning, chemicals, disinfectants,